Good afternoon, everyone. Good afternoon, everyone. I have to shake you up to wake you up. I'm Tiana Milenkovic from University of Notre Dame, and I've acted as publication chair and travel fellowship chair for this conference. Number one, let me thank you for sticking around. I know it's been 3 days of very thought-provoking and hence exhausting program. Hopefully, some sore muscles from bowling and no sore muscles from pool. It's my pleasure to introduce the last treat of the conference today, our final, last but certainly not the least keynote speaker, Dr. Mark Craven, here locally from University of Wisconsin, affiliated with 2 departments, biostatistics and medical informatics, as well as computer science. And he is also the director of NIH's Center of Excellence for Predictive Computational Phenotyping. I'm sure we're going to get a flavor of what that means, maybe a little bit throughout the day, but this center is initiative across 20 plus faculty and their research programs. So you can imagine the diversity of things that it covers. Specifically, Dr. Craven's research is in machine learning on diverse and highly dimensional data related to biology and medicine. More specifically, we will hear today about characterizing host viral interactions, but he also deals with inferring and analyzing different types of molecular data, as well as beyond molecular data, electronic health record data, and even mining literature, such as research articles using text mining and natural language processing. I'm sure there's some components of that as well, with the goal of learning new biological and medical knowledge. And without me trying to tell you what he does, let's just welcome Dr. Craven and let him explain it better. Thank you.
Thank you, Tiana, and thank you everyone for staying to the last session of the conference. And thank you to the organizers for inviting me. I'm honored to be speaking to you. And during the introduction, I guess I realized I'm not really saying much today about work that's gone on under the auspices of the Center for Predictive Computational Phenotyping. And there's been a lot of great work by many investigators, some who are in this room. So if you want to find out more about CPCP, as we call it, I suggest you check out cpcp.wisc.edu. But what I will tell you about today is a couple of projects with collaborators on campus here where we've been interested in trying to uncover host-virus interactions. And I'll talk about doing that from 2 different perspectives. So one where what's being manipulated is the host cell and finding out about host-virus interactions through that route. And the other is by looking at variation in the virus itself. So let me start by telling you about the first project. I can advance here. So it's not advancing. Let me see if I can do something. Okay, that worked. Okay, now we go. So I've had the good fortune of working with a number of great students through the years. And I think a couple of them are in the room. So in the first project I'll talk about, much of the work has been done by Debbie Chasman, who's a former PhD student, Sid Kablawi, who's on the threshold of being a former PhD student, and Matt Ziegler, who worked on this as an undergrad here. He's now a grad student at the University of Washington. And this has been work that's been in collaboration mostly with Paul Ahlquist's lab, including Brandy Ganser's in his lab, and Min-Wei Hao. And so the nature of this collaboration, I'll get to in one slide, but just for very brief background, as you probably know, viruses can't do much on their own, but instead what they do is they hijack a host cell, and throughout the life cycle of the virus, take advantage of various components of that host cell machinery in order to replicate and go on to infect other cells, right? So this figure here is giving you a sense of the various aspects of the life cycle for influenza virus in a host cell. And one way to try to gain insight into what parts of the host cell the virus is exploiting is through genome-wide suppression screens. And so Paul Ahlquist's lab was a pioneer in doing these, and the very first ones they did, I think these were maybe the very first genome-wide suppression screens for understanding virus replication, were done in yeast cells. And the basic idea, of course, is that what you can do is knock out or knock down some gene in the host cell, and then measure a phenotype of interest, such as the level of virus replication in that cell. Right? And then you go on and do this systematically and try and knock out or knock down each host cell and measure the replication phenotype. And again, the genes that I'm showing you here are yeast genes, because the first screen that we looked at was for Brome mosaic virus replicating in a yeast cell. And so the task that we've worked on is that at the end of doing one of these screens, basically what you have is a big list or a big pile of host genes that when you knock them out or knock them down, had some significant effect on the replication of the virus, right? So we can think about that as what's depicted over on the left there, where each one of those ovals I'm drawing to indicate a particular gene. Think about the color as indicating the phenotype or how replication was affected, right? We had some maybe where replication went up when we knocked out the host cell, some where it went down, somewhere where there's no effect. And then typically there's some host genes that we cannot knock out or measure replication for whatever reason. Maybe it's an essential gene. And then the task that we'd like to address is to go from that pile of genes to having hypotheses about what are the host pathways that the virus is exploiting. And moreover, how is the virus most directly interacting with host factors, and which of those host factors had an effect on the virus that was mediated through interactions of other host factors. And so, of course, one way we can address this, and there were a number of talks along these lines this morning, is to take advantage of pathway and interaction databases, right? So we can pull out interactions from various types of structured databases and then assemble what we might call a background network of known interactions that are known or thought to take place in some cell in some context. And so what I'm showing you here is the data from that very first screen we analyzed, Brome mosaic virus replicating in a yeast cell. So in this figure here, you have a little oval for each of the about 6,000 yeast genes, where the color is indicating this phenotype, right, how replication was affected when it was knocked out. And then all of the edges there are indicating various interactions that have been cataloged in yeast databases. And so we might think about this background network as giving us some insight into what gave rise to the experimentally observed data, the replication phenotypes. But of course, as in many network biology situations, if we just try and overlay the interactions on the data, we get this enormous hairball, this thing that's sometimes called a reticulome. So coming back to the task that we want to look at, right now what we're going to do is start not just from this pile of genes, but start from this background network, these interactions that may or may not play a role in replication of the virus inside that cell. And then what we'd like to do is to infer a subnetwork. That is, we'd like to prune away all of those host factors, those interactions that we think we don't have evidence are relevant to replication of that particular virus in that particular cell, and come up with hopefully a fairly, a relatively compact subnetwork, a smaller hairball. And so I'm not sure how well it's showing up here, but on the right, hopefully what you can see is that a lot of the stuff that's on the left has been grayed out, right, indicating we don't think it's relevant. And moreover, what we've done is we've placed the virus into that subnetwork. So depending on the particular context we've looked at, in some cases we have data about what parts of the host cell really have direct interactions with the virus. In other cases, we don't. In this first case of Brome mosaic virus, we had basically no data on what was directly interacting, and so that was part of the predictions that we had to make. So the way that we've approached this is to use integer linear programming, and I'll give you a gist of how we've approached this without going into a whole lot of details. But just in case you don't know, Sid Kablawi, my student on the threshold, he's deposited his thesis within days, I think. And he put together this nice slide which defines what an integer program is. And so the idea is, suppose we want to solve some problem where we're saying we want to find settings of two variables, x and y, such that we're going to minimize this, this weighted combination, 2x plus 3y. But further, suppose we have some constraints on the solutions that we want to accept, right? And so our constraints are things like x and y have to be integers, x plus y has to be greater than 7, and so on. So we have a bunch of solutions that satisfy the constraints. And then if we actually solve this as an integer program, what we would do is say that this is the particular set of assignments for the variables x and y that both satisfy the constraints and optimize the objective function, here minimizing 2x plus 3y. So we're going to use this kind of approach where now instead of having two variables, we have tens of thousands of variables that are going to indicate things like when we infer this subnetwork, for each node, for each path, for each interaction, do we want to include it, yes or no? That is, do we think it's relevant to replication of the given virus in the given cell? And so, so we just think about a little path here as shown, right? We're going to have a variable indicating, do we think this path is relevant? We have a variable for each node indicating, do we think it's relevant? In some cases, these variables are already set, right? If we have experimental evidence that node A and node C are involved in viral replication, then we'll clamp those to 1. But the others will be left free to be solved by the integer program. And then similarly, we'll have variables on the edges that we can use to decide, do we want to include those edges, those interactions in the inferred subnetwork. And then we'll have a set of constraints which are going to characterize, you know, among all the ways we can extract a subnetwork from this big background network, which of those are basically helping us account for the experimental data and seem to be consistent with what we know about biology. So the particular constraints we use have varied depending on, I would say, the particular application and to some extent what we know about the virus of the host cell. But let me just give you just a very brief sample of the types of things we might include with constraints, right? So the first one here is saying that everything that's a hit, that is every gene that when it was knocked out had a significant effect on the virus, we want to make sure that that gets included in the subnetwork. So we're going to set all of those variables to 1. We'll also have constraints which are basically saying that what we want to find is some contiguous subnetwork. So if we include a node in the network, we want to make sure that we have at least one edge that connects it to the rest of the subnetwork. So we can encode this as constraints. And then we might have other constraints saying that if we're going to include a complex in the inferred subnetwork, then at least some fraction of the proteins that make up that complex also have to have shown experimental evidence that they affect viral replication. In some cases, we've reasoned about the signs of the knockout effects and making sure that the signs are consistent with the signs of the interactions we include, and so on. And then again, the objective functions that we've used have varied somewhat, but one that we've used again and again has been based on a diffusion kernel. And the objective function is saying that we want to include nodes, that is genes in the subnetwork, not just those that we have direct experimental evidence are involved in the virus, but others as well. And we want to be a little bit generous in including those in the subnetwork. And to make the decisions about which ones are going to be included, we use this diffusion kernel to identify those that are basically in the neighborhood of lots of other hits and say those look like promising ones to include. And there's a couple of reasons why this is, I would say, a kind of a reasonable objective function. I think the real truth is that we don't really have a great objective function that tells us exactly what we want to optimize, but this is a reasonable proxy. And one reason we use this is that the initial screens we were analyzing were all based on RNAi. And there's very good evidence that the way that these RNAi screens are done and were being used result in a lot of false negatives. And so here's just a little snippet about that story. If you want to find out more, we had this meta-analysis that was published in PLOS GLBIO, I guess now almost 6 years ago, where at the time what we looked at was the 4 genome-wide screens that had been done for influenza. And so what I'm showing in the figure here is that those cryptic labels, DL1, U2OS, those are indicating the different screens, basically naming a cell line, and the subscript indicates a country in which that assay was done where that distinguishes between the same cell line. So we have these 4 studies, and then the bars are showing the number of hits identified by each study, right? So that is the group of 4 bars here, right? So each screen identified somewhere between 100 and 200 genes as being directly relevant. And then these bars here are showing genes that were identified by pairs of studies, right? So you can read this by looking at some bar and then looking down to see where there are 2 pluses, right? And this bar indicates that these first 2 screens identified 2 genes in common. If we work our way over to the very last part here, this is showing that among these 4 screens that collectively identified about 600 genes as being relevant to influenza replication, the intersection was exactly 1 gene, right? So when we looked at this data, it wasn't clear, is this happening because these screens have lots and lots of false positives or because they have lots of false negatives? And we looked at this in a number of ways. By looking at pathway level agreement, by looking at interactions, and probably most importantly by using a stochastic model developed by Michael Newton that tried to explain in detail all the experimental data. And the upshot of this is that these screens have a low degree of agreement because they each have a lot of false negatives, and each one is really just gathering a snippet of the host subnetwork that's really involved in influenza replication. And so what the figure on the right here is showing is that if you kind of project out and you did more and more screens that had about the same degree of dependence as the 4 that were done to each other, that what we're projecting is that you'd identify somewhere between 2,000 and 3,000 host genes that really have some effect on influenza replication. So this is a bit of an aside, but anyway, it's one motivation for that idea of using the diffusion kernel to try and gather up a number of nodes in the subnetwork that the experimental screens did not directly identify as being involved in the virus replication. Okay. And then a last key element of our approach here is that our optimization problem is basically under constraint. And we can find quite a number of subnetworks that are either optimal or close to optimal. And so what we do when we run this approach is we actually subsample the data a bunch of times, infer subnetworks from each subsample, and then ensemble them and look to see, as we look at these different subsamples of the data, what properties of the inferred subnetworks seem to be conserved across a lot of those subsamples. So coming back to this figure I showed you before, where we're starting with the background network for yeast and the Brome mosaic virus data, if we take this approach and apply it to this case, then the reduction in the hairball is something like from this to this, right? Where this is the inferred subnetwork, or the consensus one, where we had a fair degree of confidence in the nodes shown here and the interactions shown here. And moreover, in this case, we're also making predictions about which of those host factors are most proximally interacting with the virus. So we can always infer something like this, right? No matter if we could provide complete noise as input, and the method is going to give us back a subnetwork. So how do we know if this actually corresponds to any underlying biological reality? And one way we've done that is to hold aside some of the phenotype data. That is, for some of the genes, right, pretend like we didn't get the measurements about whether those genes were involved in virus replication or not. Let's hide that data. Let's infer a subnetwork and see if the inferred subnetwork includes those genes for which we held the data where the experimental data said they should be included. And let's see if we exclude those genes where we hid the data where the experimental data suggested they should not be included. And so what I'll show you is the results, not for doing this with Brown mosaic virus, which the results look largely the same. But this is a case where we're looking at data in human cells for HIV replication, where the data is coming from 5 different genome-wide screens for HIV. And so what we can do is look at these predictions of whether we think a gene is relevant or not against the gold standard of what do the experimental screens say. And so the way that we're viewing this here is in terms of a precision recall curve, right? Where the notion of a true positive is we predict a gene should be included in the relevant subnetwork, and the experimental data said, yes, it was identified in a screen as being relevant. So in this figure here, the horizontal line is showing you the default level of precision, right? If we predicted that every gene was truly involved in HIV replication, what fraction of the time would we be correct according to the experimental data? And then the curve there is showing this curve we get from our inferred subnetworks, where we're making predictions about what's being involved. So clearly you can see that there's some predictive value, right? These subnetworks are certainly not completely accurate. Maybe huge parts of them are wrong, but they're capturing something about the underlying host processes that are being exploited by HIV. In some cases also, we can dig down into parts of these subnetworks and ask, well, do the predictions they make accord with other experiments that we didn't use in the analysis? And so here's one anecdote along these lines. This is coming from the analysis of BMV in yeast, where what this is kind of cluttered, but what this figure is showing you is that it's a little part of the subnetwork that's saying that we had experimental evidence that Bro1 was involved in BMV replication, right? Because it's shaded in. And that this part of the subnetwork is predicting that Bro1 has its effect on viral replication through VPS4 and SNF7, which are part of the escort3 complex. But those guys are not shaded in because we did not have any direct experimental measurements about whether they're involved or not. Right? This is all pure prediction. And it turns out that this is a prediction that was verified by independent experiments in the Alquist lab, right, where they know that VPS4 and SNF7 really are involved in virus replication. This is happening through the escort pathway, which is involved in membrane scission, where the replication is actually happening. So more recently, we've applied this approach to the same kind of genome-wide screen data for a number of other viruses. So those results with a similar type of experimental evaluation, or computational evaluation, are being shown in this slide here for 5 different viruses. So again, in each case, what I'm showing you here is the precision recall curve we get from the predictions made by our inferred subnetworks. Those are shown in red. The blue curves are showing what if we do something similar where we don't use the whole IP, but we just use a diffusion kernel to rank genes and use that diffusion kernel to predict which genes we think are involved in virus replication. So a couple of things you can see here is that one, for some viruses, again, we do quite well. These models seem to be capturing something about the real underlying biology, as indicated by the predictions being having reasonable predictive signal. In many cases, we're doing better than the diffusion kernel, right? So the IP, which is inferring this subnetwork, is adding some predictive value that goes beyond just what the diffusion kernel is doing. But then there's cases like West Nile and Zika where the predictive value of these subnetworks is not so great. And I have to tell you, we're not quite sure why that is to date. I mean, we can see there is some predictive value there because the PR curves are above that default line you would get if we just predicted that everything was involved. But again, we're not getting nearly as much signal as we are for some of the other viruses. So one limitation with this whole approach is that we know there's a lot of incompleteness. And this incompleteness comes in a couple of different forms. So one is we know the background networks are highly incomplete, right? Just because there's lots of interactions, lots of things happening in the host cells, that haven't been cataloged in the various structured databases that we're tapping into to assemble these background networks. Right. Moreover, we know that there's a lot of direct host virus interactions that have not been cataloged on databases where we're pulling those from. And as I mentioned, we know that the genome-wide screen data is also incomplete, right? And is not capturing all of the host factors that may be involved in virus replication. And so something that we've spent a fair amount of effort on is asking, can we address these limitations by thinking about the scientific literature as another source of evidence that we can use in this kind of analysis? And of course, one way you could use the scientific literature is to read all of it and then incorporate that into the models and such. But we're interested in doing this by using automated methods. And there's a couple of ways that we've looked at approaching this. So one fairly simple way is just to look at kind of co-occurrence statistics. So for example, if we wanted to find out, do some simple analysis of the literature and ask what human genes seem to be involved in HIV replication, then we might look for surprising number of co-occurrences between mentions of human genes and concepts like HIV replication, right, as we see in this article here. So we have a tool which can do this. It's not at all tailored towards trying to elicit information about host virus interactions, but it's a tool called GADGET that what you can do is issue basically an arbitrary query, right? Almost any query you would give to PubMed. And whereas if you issue that query to PubMed, PubMed would give you back a ranked list of articles. Instead, what GADGET will do is give you back a ranked list of genes, or in another search mode, a ranked list of metabolites, where it's ranking them according to what it thinks it's the degree of association of that entity with the query is. So what I'm showing you here is a snippet of a screenshot where we issued a query on HIV replication, and it's showing you the first few hits, that is the genes that it ranks most highly, at least according to one criterion. One criterion that's trying to capture to what extent are these genes, do they seem to be occurring with HIV replication to a surprising degree. So this is one tool we've used where what we can do is issue this kind of query, HIV replication, see what genes are identified by GADGET according to that query, and now effectively treat this as if it were another experimental screen that's looking at HIV replication and using it as we use our experimental screens. So here's an experiment where we did just that. Again, this is looking at HIV, inferring subnetworks that are trying to capture the host machinery that's involved in HIV replication. Again, we're looking at the same kind of analysis where we're using these subnetworks to predict which host genes are involved in that replication and using experimental evidence as our gold standard to evaluate these predictions. So the blue precision recall curve being shown here is the same one you saw before, where we inferred those subnetworks just with the experimental evidence. The red curve is what if we use those 5 experimental screens plus use these hits we get from GADGET and treat that as if it were another experimental screen, but where our validation is just looking at experimental data to ask, are genes truly involved? And so what you can see here is we get a nice boost in precision, at least at the low recall, high confidence end of our precision recall curve by using the literature here. So this is a fairly simple, but seems to be a fairly effective way of identifying genes that are associated with almost any kind of process we might query on. But we've also looked at doing deeper analysis of literature by pulling out sentences, parsing sentences, taking representations of those parsed sentences, and then training information extraction models to try to identify specific relations. Right? So one of those models that we've trained, for example, is looking at co-occurrences of host proteins and virus proteins, and now trying to decide, is that co-occurrence saying that one of those proteins is involved in a degradation relationship with the other protein? I'm saying protein here, but it could be the messenger RNA as well, as the sentence indicates. Right? And so we have models that can look at these co-occurrences and now try to say, is that an instance of some particular relation of interest? If we think we have enough evidence that it is, and if our model's good, then we can put that interaction into the background network and use it as part of the subnetwork inference process. Right? So now looking at using kind of both of these ways of tapping into the literature, let's come back to a couple of these other screens. So I'm showing you again a screen for hepatitis C, where we get curves that look pretty good, one for West Nile where the curves are not so great. But what we can see here is the value that we seem to be getting by using literature as another source of evidence. So in both cases here, the red precision recall curve is showing what happens if we just use the experimental data in inferring these subnetworks. The blue curve is showing what if we use the experimental data plus the literature extracted information. So that's taking two forms here, both hits, that is genes that we think are involved in the virus replication, as well as direct host virus interactions that we think are part of that replication process. And so what we can see in both cases is we get some boost in precision by using the literature, and moreover, we get a boost in the endpoint recall by using the literature. And the recall in the case of West Nile is almost doubled by using the literature. So the story is not quite always so nice as this. In some cases, we add all this literature extracted information in, and it doesn't help at all, but it rarely seems to hurt in terms of the predictive accuracy. So that's kind of one side of our work in looking at host virus interactions, where again, we're looking at perturbing the host cell and seeing what effect that has on the virus. So the other project that I'll tell you about is now looking at kind of holding the host constant, but looking at variation in the virus and trying to understand what that tells us about how the virus is responsible for disease phenotypes. So this project has been joint with Curtis Brandt's lab. So Curtis is in the Department of Ophthalmology here at Wisconsin. And we've worked closely with Aaron Kolb, who's a scientist in his lab. And then Cuban Lee is a former grad student who did really all the work on this project, or all the computational work. So the nature of the experiments that I'll be talking about here, again, are ones where we're looking at varying the viral genome. And in this case, they're studying how HSV causes eye disease. So what they do is they can take a particular strain of the virus, infect mice. They then follow those mice over a time course of several weeks. And over that time course, they're then tracking and recording various dimensions of eye disease phenotypes that are caused by the virus. And so now we can get this association between viral genome and disease phenotypes that it causes. And I know we're after lunch and after snack break, so I won't show you pictures of the eye disease phenotypes, but I'll use this representation here, where we have just these ovals where the color intensity you could think about is indicating the severity of the disease phenotype caused by that particular virus strain. So another way to think about the data, and one that's maybe a little more informative, is that HSV has a genome of about 160 kilobases. So it's a pretty large genome for a virus. And what Curtis's lab has done is isolated a number of different strains. And in this experiment, what they did is they generated different recombinants. So we started with two parental strains of HSV. You can think about one is the red virus, the other is the white virus. And then they generated recombinants, both in vitro and in vivo. And so each one of these recombinants now is going to be a mosaic of the two parental strains. So each one of these bars here you can think of as a depiction of a particular viral genome, where the red components of that bar are indicating segments of that genome that have come from the red parental strain. The white bars are indicating segments that have come from the white parental strain. And then the purple bars are indicating components that have come, or components where we can't really determine whether it's come from the red or the white. And then the thicker part of each bar is indicating the parts of the genome that are highly repetitive. And that's basically where we get these ambiguous regions that we can't determine. So then we have whole genome sequences for these strains. We've done a multiple sequence alignment. We've infected mice. We've measured these eye disease phenotypes then over several weeks. And we have about 5 different phenotypes that are measured for each infected animal. So the computational task that we've addressed then is, can we take these whole genome sequences as well as these disease phenotype measurements, again, in a host animal, and can we identify the genetic determinants of the disease phenotypes? What are the regions of the viral genome that seem to be associated with severity of the disease? And so the way that we frame this problem is that we're starting from about 70 different viral genomes. We can align the genomes, as I said. We can then identify different haplotype blocks, right, where we can say for each one of these strains that we have a high degree of certainty that this particular strain is inherited a given region, either from one parent or the other. So for the given data set we have, we wind up with about 547 of these haplotype blocks. So then we can represent any given strain as a vector of these haplotype blocks, right, and which variant it has in that block. And we can learn a model to map from that vector of 547 elements to a numerical disease phenotype. And we've used fairly standard approaches to learn these models. So this is a regression task since we have numeric disease phenotypes. So we've learned some models that are linear models where we've used regularized regression methods like lasso and ridge regression. And we've also learned random forest models, which are able to capture more nonlinear and interactive relationships between the viral genome and the disease phenotypes. So we've evaluated these models by asking if we do leave one out cross-validation, that is, if we hold aside the data we have for a given viral strain, learn these models with the rest of the data, how well can we predict the disease phenotype for the held aside strain? So what I'm showing you in the figure here is results from that kind of leave one out cross-validation experiment, where we're looking at now models that have been learned for 5 different disease phenotypes. So weight loss, mean peak titer, vascularization, et cetera. And along the x-axis here, what we're looking at is a measure of R squared between the predicted phenotype and the actual measured phenotype. So the red dots there are showing you, in this case, what were the R squared measurements for models learned for this task using random forest. So what you can see at the R squared values are mostly in the range of about 0.4 to 0.6, right, indicating that we're capturing a fair degree of association between variation in the viral genome and the induced disease phenotypes, but obviously not all of the variation there. The green dots are showing what these numbers look like when we learn models at a time when we had just 40 different strains as opposed to about 70 different strains. And so as you can see, as we've gotten more data, the models have captured more about the relationship between the viral genotype and the disease phenotype. And then the blue bar and whisker plots are showing what do these R squared values look like if we randomize our phenotype data. Right? That is, we keep intact all of our feature vectors representing the genomes of the viral genotypes. We now randomize the phenotypes, but learn the models, predict phenotypes for the held out data. Okay, and so what you can see, not surprisingly, is that the R squared values for the randomized data are not very good, and they're even below 0, because these models now can overfit instead of just doing as well as predicting the mean. So from this, we conclude that the models are capturing a fair bit of the information about that the genotype carries about the disease severity caused by the various viral strains. But what we're really interested in doing is not making predictions, but now asking what do those models tell us about what are the viral genetic determinants of eye disease. And what I showed you was the R-squared values for random forest models. For some of the phenotypes, we got models that were just as good using a linear model, in particular ridge regression. But for some of the phenotypes, the best models were random forests. And one limitation of random forests is like any other kind of ensemble model, they're basically black boxes. Right? These random forests have thousands of trees in them. It's hard to identify what are really the important variables. And moreover, another question we want to ask is not just what seem to be the important variables or the important haplotypes, but how finally can we resolve that in the genome and confidently say that it really is this haplotype as opposed to some neighboring haplotype, which is just highly linked with that one. So we'd like to be able to peer inside the black box. And again, I'm not going to say a lot about this today, but also in the case of a random forest, say not just what are the important variables, but if there are important interactions among those variables. Right? The models seem to be capturing some kind of epistatic interactions. What are they? And so this is a problem that we're interested in more generally. And this is actually a problem that's of high interest now in the machine learning community, especially with a lot of successes due to deep networks. And when I say this problem, I mean this problem of having a learned model which has some kind of good predictive value, but is a black box that's hard to interpret. Right? How can we gain some understanding into this model? And so we're quite interested in this particular problem. And we're interested in methods which satisfy a number of different criteria that are listed here that I won't go into detail. But given the time, what I think I'll do is move ahead and tell you about how we've applied these methods to this particular case of looking at random forest models that are mapping viral genotypes to host disease phenotypes. Okay. And so the basis for this approach is that what we can do, of course, is we can take a learned model and we can take some feature vector and ask, how good was the prediction of the model for that feature vector? And then we can perturb that vector. Right? Maybe we can take some haplotype value, swap that out, and put in another haplotype and ask how would that have affected the prediction of the model? If it has a big effect, then it suggests the model thinks that that particular haplotype block is important. Okay? So we can do these kind of perturbations and we can compare the loss or the error of the model before we did the perturbation and after, and that's going to tell us something about how important a feature is. So this isn't a new idea. This has been done quite a bit, and in particular, quite a bit with random forests. But something we want to be able to do is to be able to say, not just be able to rank these features or haplotypes according to their importance, but be able to say, statistically, can we say it really had a significant effect on the loss? And moreover, I said we want to be able to identify at what resolution can we really say that this haplotype or this segment of haplotypes seems to contain some kind of variant that's important to the disease phenotype. So again, if we do these perturbations, something we can do is we can always do hypothesis testing. We can ask, for each instance we have, what was the loss or what was the error of the prediction made by the model for that instance before we did a perturbation and then after. And then we can do some kind of hypothesis test to see when we perturb this feature, did it seem to have a significant effect on the model's loss? But in many domains, we have not just features, but we have meaningful hierarchies or aggregations over features. And so this is something that we've tried to take advantage of. And the way we can take advantage of this is to think about perturbing not just individual features, but whole groups of features that may correspond to some intermediate node in a hierarchy. So the hierarchy here I'm showing you is part of the ICD-10 hierarchy of diseases, but in just a minute I'll tell you about how we can map this onto this idea of haplotypes and groups of haplotypes. But the basic operation we're going to do is now, instead of necessarily looking at individual features, perturbing them and asking what was the effect on the model's prediction, we can look at a whole group of features, perturb that group, and ask how did that affect the model's prediction. And when I say perturb here, what I mean is we might take, for a given instance, some sequence of haplotype blocks, pull that out of the instance, and now randomly pluck in another sequence of haplotype blocks, say from another viral strain. And there's this nice method for if you have a hierarchy and you're doing lots of hypothesis tests, being able to do false discovery rate control in the hierarchy. So this is due to Yekutieli. And so this is something that we're going to leverage in this analysis, where the idea is we're going to start with a hierarchy over all of our features. Right? So think about our features as being haplotype blocks. Up at the root node, we'll be representing basically the whole genome of the virus. And as we go down, we'll be looking at segments of the viral genome. And so what we can do is we can start up at the root node. We can say, what if we perturb the whole virus, the whole feature vector for some virus? Will that affect our model's prediction? Right? And in that case, hopefully it will, because it's basically saying if we randomize our input, our viral genotype, will that have an effect on the model's prediction? It should. If it does, and we conclude it has some statistically significant effect, then what we can do is move down the hierarchy and test other feature groups. Okay, so we might move down to now this node here, where we're going to say, let's take a whole group of features, say up to haplotype J, and let's perturb that whole group and see, does that perturbation have a significant effect on the predictions of the model? If it does, we'll keep moving down the hierarchy. So in this case, suppose we perturb that, we see it doesn't have a statistically significant effect on the model's predictions, then what we note that what we can do is not move further down that part of the hierarchy, but instead move over to some other node and now test this other group of features. And again, if we test that, we see it has a significant effect, then we would move further down the hierarchy. So again, the advantage of doing this in a hierarchy is we can gain greater power to detect what's really important. And moreover, we can figure out at what resolution can we say that some region of the genome seems to contain some variant or variants that have an effect on the disease phenotype. So here's an analysis of doing this now for these models that are mapping HSV genotype to host disease phenotype. In this case, we're looking at just one of the phenotypes. So the hierarchy now is going to represent groups of these haplotypes. So the root node is representing the whole genome. One of the leaf nodes that's shown as a square block is representing a specific haplotype. The oval nodes are representing groups of contiguous haplotypes. And then the color intensity for each node here is indicating basically a p-value. So when we knock that guy out or perturb that, how much effect did it have on the model's predictions? So a couple things that we can see in this figure here. So one is that in some cases, we can move all the way down to one of these haplotypes. That is one of the base features given to the model, and say that that feature seems to have a statistically significant effect on this model's predictions, right? Suggesting that that haplotype really contains some variation that's important for determining the disease phenotype. But in other cases, these nodes we come down to where we have to stop are not individual haplotypes, but they're sequences of adjacent haplotypes, right? So the upshot here is that what we're able to do is say that somewhere in this sequence of haplotypes, there's some variant that's important, but we can't really localize it to one haplotype block, presumably because these haplotypes are too correlated, right? Too linked to be able to tell which one is important. And then the other thing to note is that there's whole parts of this hierarchy subtree that we can prune away without having to test. Because we tested some parental node, we said when we perturbed all of the haplotypes under that parent node, it had no significant effect on the predictions of the model. Okay. So another way to look at this is we can now, again, start from one of these black box random forest models. And I think I'll skip over this, but just show you this figure here. we can then map it down to the viral genome coordinates. So that's what's being shown along the bottom part of the figure here. Again, the viral genome is about 160 kilobases. And we can ask, where in that genome are we finding variants that seem to have an association with the disease phenotype? And here what we're doing is we're marking where those variants are, color coding them according to a p-value. So the top bar there is showing you this analysis for one phenotype, and here's what it looks like for two other disease phenotypes caused by HSV. Right, so you can see a couple of things, right? One is that like right in the middle, we get this really strong island of pathogenicity, which seems to be linked with all three phenotypes. The first 80 to 90 kb of the genome don't seem to matter much for pathogenicity for any of the three phenotypes. So just to wrap up, maybe I'll switch away a bit from host-virus interactions and very briefly tell you about how we've applied this method in a quite different context. So this is a context where we're looking not at host-virus data, at least in the anecdote I'll tell you about, but where we're looking at data from electronic health records. So we have data on individual subjects where we can look at their clinical histories as represented in their electronic health record, as well as demographic data. And the task we're looking at is, given we have someone who's an asthma patient, and we can observe their clinical history up to some point in time, then can we predict whether they'll have a severe asthma attack within the next few months? Okay, so this is the task that we're looking at. And this is a task where we've learned models from patient histories, and we've validated those models by making predictions for held-aside patient histories. And one of the models we've learned there is a complicated black box. So it's a type of neural network called an LSTM. So basically a neural network that is looking at the temporal data we see in the electronic health record. And at each time step, it's taking in a representation of that data indicating on each encounter that patient had what kinds of diagnoses were recorded, what kinds of procedures were done, were there certain prescriptions, what kind of vitals were recorded, and so on. And then given this history for a patient, the model then outputs a prediction, which we interpret as a probability that they're going to have a severe asthma attack within the next 90 days. Okay, so we have this model, which again is really a black box. So it makes reasonable predictions, but it's really hard to look into that model and ask what features are important, what interactions are important. As in the case I just told you about, there's also a lot of structure over the features, right? So for diagnoses, we have this ICD-9 hierarchy, which describes relatedness among those diagnoses. So this same kind of hierarchical procedure we can use now to analyze this model and ask what groups of features really seem to be important for making predictions in that model. And so here's a little snippet of that analysis where what you're looking at is kind of the top part of this ICD-9 hierarchy of different diagnoses. Here we're showing it just in terms of code, so you probably can't read anything. And this is showing you kind of only a small part of the top part of the ICD-9. This really has thousands of nodes. But if we drill down into this and ask, well, which of these recent diagnoses, when they occur in a patient's record, seem to be important for predicting asthma exacerbations, then we can see the kinds of things that we hope to see. Like in this case, what we're looking at here is that there's this whole group of nodes here which have to do with respiratory diagnoses. Right? So if a patient has a lot of recent respiratory diagnoses entered, that probably indicates that they're struggling with their asthma, they're at high risk for asthma exacerbations. Right? And right down there at the base node, we can see that a code of 493, which indicates asthma diagnosis being recorded as a problem, right? That's a very good predictor. But then we see things that are less surprising. So we also see that if there are recent diagnoses of mental health disorders, that seems to be a risk factor also for having severe asthma attacks. And in this is a case where we can't go down and pin this down to very specific diagnoses of mental health disorders. It's more the case that this whole group of disorders, if we see things there, seems to be important. And there's literature that supports why this might be the case. right? That basically the bottom line is that people with mental health disorders often struggle with doing the routine maintenance they need to do to keep their asthma under control. So just to wrap up and to bring things back to host-virus interactions, the messages I tried to convey today are several-fold. So one, not surprisingly, I would argue that we can gain insight into host-virus interactions by looking at really a diversity of data types, right? So experimental data by tapping into structured databases, by extracting information from literature, by manipulating both host cells and viral genomes. That the models we learn in these cases, I think, have value in a couple of ways. So they can help interpret what we've seen in these genome-wide screens and why host factors might play a role in virus replication. They can make predictions about other host factors that the screens didn't turn up that seem to be implicated in host or in virus replication in the host. They can suggest what are the genetic determinants in the virus of a disease process, such as viral infection. Something else I tried to convey is that I think it's important to think about the scientific literature as another source of evidence, and that ways to tap into the scientific literature automatically can have value for studies like these. And then finally, that in many cases, even if we care about interpretability, it may still be feasible to learn some kind of black box model and then analyze that black box to get some kind of insight into what it says about important features, groups of features, or interactions among those features. And then finally, let me just acknowledge funding sources that have played a role in the work I've talked about. So I think I went longer than I was supposed to, but I'd be happy to take more questions. And after the conference, I'll be around. There's actually good beer downstairs. I'd be happy to buy beer for anyone who wants to stay and ask more questions later. Okay.
On the image you showed of the different features for the virus with the different color codings, you seemed in a couple of cases to gain significance from one higher level going down to a lower level. And intuitively, to me, that seems like it shouldn't happen. Obviously it does. So
Yeah.
But can you explain that?
So I can tell you one reason that might happen anyway. So we might have a group of features where there's, say, one feature in that group that really is highly predictive. Right? So we'd expect to have a strong p-value if we just analyze that feature. But maybe there's some neighboring features that have been used in the model, but where the model has done a bit of overfitting. And so they almost have negative predictive value. So now collectively, if you look at the predictive value of the set of features, right, the high predictive value of this one feature may be getting drowned out a little bit by those that really have poor predictive value. So I think that's why we sometimes see this kind of not exactly what you might expect in terms of increasing intensity as you go down, or decreasing intensity as you go down.
I have two very nice technical questions. So the first part of your modeling the nodes, which is the gene, the edge, which is probably some sort of action, the inference of the genes. And you seem to show one slide showing the example of the Y value I recall is which of the node X is the edge. So I think you showed one slide, but none of those are the values are integer. So Y is some numbers, X is also numbers. Am I just misinterpreting your integer programming? The second question is that your so how do you? That's a feature in your model, in your machine learning training input. So you just modeling those as 0 and 1, which of them?
Yes. Okay. So taking the first question first. So in the integer program, these X and Y variables are indicating whether we're going to include some edge or include some node in the inferred subnetwork. So those are integer variables that take on values of 0 or 1, right? Don't include or do include. I think what might have been confusing is I showed you a slide where we had something like x equals 0.99. And so really see why you got confused now. So what that was indicating was a degree of confidence. That was indicating that in this ensemble of, say, 100 subnetworks, that edge was included 99 times. So that's where the non-integer came from. Coming to the haplotype question, since in the strains that we're analyzing here, they're all recombinants from 2 parental strains, that the haplotype blocks are taking on one of 2 values. So those are 0, 1s. What we also see are in various places are novel mutations, right? We see bases that are not present in either the parental strains. And for now, we're not really looking at those because we don't have enough strains to really be able to get any kind of enough of a signal to do much with those.
One last question.
Consider incorporating like spatial or temporal information features by other towards importance of subsequent special features?
Yeah, that's a great question. So we've certainly considered it, and I think that's a wide open area, right? How can you better incorporate spatial temporal aspects of these whole processes? And we've done almost nothing along those lines yet. I won't say nothing because the one thing that we've done that I didn't tell you about is another way that we've looked at using the literature is not to influence how we come up with the subnetwork, but once we have a subnetwork, how can we look at what we call different views of it? So different parts of the subnetwork that may seem to be tied to different themes. And so in that case, at times we've looked at localization data. Right? So we might ask what parts of this seem to be involved in membrane-related processes. But we've not really looked at all at taking into account cellular compartments and maybe even larger scale spatial considerations and using that kind of information in inferring subnetworks. I think that's a great thing to delve into.
For any remaining questions, feel free to take up Dr. Craven on his offer to buy you some beer. So let's thank our keynote speaker again.
Thank you.